In our previous lecture, we discussed about how glucose was converted into pyruvate, and now we will proceed further. When glucose is converted into pyruvate, it is then converted into acetyl coenzyme. Acetyl coenzyme. This acetyl coenzyme will react with oxaloacetate. Oxaloacetate. This is a compound which is already present. This is a compound which is already present in the cell. Oxaloacetate will combine with acetyl coenzyme, and these two will collectively form another compound which is called citrate. So basically we have started our Kreb cycle, which is the second step of ATP synthesis. In our previous lecture we discussed how glucose was converted into pyruvate, and in this lecture we will discuss the next proceeding steps, that how these pyruvate will generate ATPs. Pyruvate is converted into acetyl coenzyme, and this acetyl coenzyme will react with oxaloacetate, a compound which is already present in a cell, and they will form a new compound which is called citrate. This citrate is converted into isocitrate. This isocitrate is then converted into alpha ketoglutarate. This alpha ketoglutarate will react with succinyl coenzyme and will be converted into succinate. This succinate compound will be converted into fumarate, then malate, and back to oxaloacetate. Now, what are the enzymes which will take part in the formation of these respective compound? We will discuss those enzymes by the help of a mnemonic. I have made a mnemonic for our easy learning, which is PC add TS FM. PC add TS FM. So this is a mnemonic for all those enzymes which will take part in the formation of these products. The first symbol of this mnemonic is P. P stands for pyruvate dehydrogenase. Pyruvate dehydrogenase. This enzyme will act over here. Pyruvate dehydrogenase. This enzyme will act over here and will convert this pyruvate into acetyl coenzyme. This acetyl coenzyme along with oxaloacetate will be converted into citrate by means of the other enzyme, which is C. C stands for citrate synthetase. Citrate synthetase. This enzyme will help in conversion of acetyl coenzyme and oxaloacetate into citrate. The other enzyme with the symbol A, which is aconitase. Aconitase. This enzyme will convert citrate into isocitrate. This isocitrate will be then converted into alpha ketoglutarate by the help of another enzyme, which I have used the symbol D. D stand for dehydrogenase. Dehydrogenase. And what it gonna dehydrogenase? This isocitrate. So isocitrate dehydrogenase. This enzyme will convert this isocitrate into alpha ketoglutarate. The other symbol is again D, which means there will again a dehydrogenase procedure. So the next enzyme would be alpha keto dehydrogenase. This will convert this alpha ketoglutarate by the help of succinyl coenzyme into succinate. So here another enzyme will react, which we have used the symbol T, which is thiakinase. Thiakinase. and this succinate will be formed. This succinate will be converted into fumarate by the help of another enzyme which we have used mnemonic S, which means succinate dehydrogenase. Now F, which mean another enzyme which mean fumarase. This enzyme will convert this fumarate into malate. This malate will then be converted into oxaloacetate. Last symbol is M, which means it would be malate dehydrogenase. Malate dehydrogenase. So this was all about the Kreb cycle. We discussed about the various compound which is formed, which was glucose which was converted into pyruvate in glycolysis. This was a procedure which was taken place in the process of glycolysis. Then in Kreb cycle we begin with pyruvate conversion into acetyl coenzyme, which along with oxaloacetate was converted into citrate, and then isocitrate, then alpha ketoglutarate. Then this alpha ketoglutarate along with succinyl coenzyme was converted into succinate, then fumarate. Then this fumarate was converted into malate, and then oxaloacetate. This whole procedure we will look how energy is basically released. So this isocitrate, which means it has basically 6 carbons. 1, 2, 3, 4, 5, 6. In isocitrate compound there were 6 carbon, and when it was converted into alpha ketoglutarate, there was release of carbon dioxide, which means this isocitrate, this 6 compound was converted into how many compounds? 5 carbon atoms. One of the carbon was released along with this oxygen and carbon dioxide. So this alpha ketoglutarate has 5 carbons. In isocitrate and before all these steps we had 6 carbons. This was converted into 5 carbon with release of this carbon dioxide. Here NAD was converted into NADH. Remember, this is a very important compound and it would be used in electron transport chain in generation of energy. When this alpha ketoglutarate converted into succinyl coenzyme, there was again release of carbon dioxide, and again NAD converted into NADH. So till now we have 2 NADH. Next, when succinyl coenzyme converted into succinate, there in this step ADP is converted into ATP. So here we have a molecule of energy ATP. When succinyl coenzyme converted into succinate, ADP was converted into ATP. When succinate converted into fumarate, FAD converted into FADH. And when fumarate converted into malate, there is addition of water which convert this malate into oxaloacetate. Again there is release of NAD into NADH. So here you can see 1, 2, 3 NADH. There is release of ATP molecule in this step, succinate into fumarate, release of FAD into FADH. One FADH molecule was formed. There is one ADP, and here you can see 3 NADPH. So now let's conclude the whole lecture by how many amounts of ATP, NADH and FADH formed. So ATP produced in the whole TCA cycle, which is also called citric acid cycle, there was the formation of 3 NADH, 9 ATP released, 1 FADH, 2 ATP released, 1 GTP, 1 ATP release. So net ATP produced in the whole citric acid cycle was 12 ATP, 12 ATP.